I want to point out that this is a collaboration with Denise Brusewitz and Whitney King. I think I saw come in. There he is. Up at Colby College. Bigelow has a partnership with Colby. We do teaching alongside our colleagues up at Colby, and we're trying to increase research collaborations as well. And so I first started working in freshwater systems through the Belgrades and in collaboration with Denise and Whitney, looking at an organism called Gliotrichia, which many of you might be familiar with. It's a cyanobacteria that can bloom to relatively high abundances, even in our oligotrophic clean water lakes. This is not a picture from Maine, but this is sort of a preview of a movie that I have at the very end of the talk, showing a Gliotrichia bloom right at the surface underneath a dock over in New Hampshire, where I grew up. And so this was actually at the under the dock of one of our board members, and he had invited me over to check out the algae, what was growing, or actually even plants, I guess, what was growing in his little bay. And as soon as we walked out onto his dock, we noticed this huge bloom of Gliotrichia going on. So that sort of drove our sampling for that particular day. But I'll start off just by giving you a little bit of background for those of you who might not know much about cyanobacteria. They're evolutionarily an ancient group. They date back billions of years. There are fossils of cyanobacteria. This is sort of the evolution of life on Earth, back to about 3.5 billion years ago. And they're an incredibly complex group just because they've had eons to evolve and diversify. And they've branched into many, many different groups, and many of these groups are of concern because a lot of them produce toxins. And not just one toxin, but dozens of toxins. And so this is why we need to take them into consideration in terms of lake water quality sampling. This is what cyanobacteria look like. Many of them have this kind of cylindrical shape, and you'll notice that some of them are broken up by these little sort of rounder spheres in the chain. And these are often sites where the cyanobacteria fix nitrogen. So they don't even need a nitrogen source in the water, an inorganic nitrogen source. They can take dissolved atmospheric nitrogen and convert that into an inorganic form, which they can use for growth. So a lot of it comes down to phosphate dynamics in lakes, as many people probably know. Since they can essentially synthesize all the nitrogen they need for growth, it really becomes a phosphate story. A lot of them, again, are these sort of filamentous types, but you can have colony-forming types that form spheres. This in particular is microcystis. That's a particularly nasty cyanobacterium, produces a lot of different toxins. These are all images from coastal California in these little sort of freshwater ecosystems along the coast of Southern California in particular. So there's a huge amount of diversity, even in areas with not many lakes. Again, just to hit home on their diversity, this is a really complex graph. It shows some taxonomic relationships, but it also what I really want to point out is that this row indicates the habitat, and the habitat can range from marine in the dark blue dots, so all these dark blue dots indicate marine species. The light blue on this lower row is our freshwater species. Cyanobacteria can be found in soils. They can be aerosolized and blown around in the wind. All of this layer here that ranges sort of from clear to green indicates whether they can fix atmospheric nitrogen. So all the green dots indicate the groups that don't need an inorganic nitrogen source. Some of them are even thermophilic and can be found in these hot water ecosystems. And then, let's see, what else are we looking at? There's a degree of genomic similarity. All of these sort of lines sort of attaching between the different branches of this phylogenetic tree indicate areas where they've exchanged genes with one another. So there's a high degree of genetic transfer in the microbial world. And so a lot of a lot of these groups, you'll find little bits of what look like other organisms in their genome. And this is one way that again, these groups diversify and can find a way to make a living in so many different types of environments. Okay, so what good is pond scum? So I'll focus a little bit about on the positive side of cyanobacteria. I don't want to completely downplay them, but because they are so evolutionarily diverse, they've existed for such a long time on Earth, there's a huge amount of biotech potential in cyanobacteria. One thing we do at Bigelow Laboratory is we maintain a living culture collection of both marine and, to a lesser extent, freshwater algal species. There are lots of bioactive compounds. They produce, again, there are some that produce anti-cancer drugs and may even be a source of antibiotics. This is actually a farm, a cyanobacterial farm out in Hawaii. Wow. On the Big Island. And I've toured this facility and it's impressive. It's 60 acres of 10 centimeter deep ponds where they're purposely growing cyanobacteria to add to your smoothies as supplements, so as a protein source. So if you ever see that, and these green tea health beverages, they'll have a cyanobacterial source, and it's grown probably in this pond. Of course, there was a lot of effort kind of in the early part of the 2000s to look at the possibility of deriving biofuels from algae, because they do grow so rapidly, and many of them do concentrate lipids, or they have a high degree of lipid content. They can be extracted to produce a fuel. That's just a close-up of the facility on the Big Island. So pretty amazing that there's 60 acres of cyanobacteria being grown on purpose. Okay, so the problem with cyanobacteria, and just to point out, we call these blooms, when they become so dense like this, cyanohabs. So you heard the term harmful algal bloom. Typically, algae is reserved for eukaryotic microorganisms, whereas these are bacteria that happen to have pigments and behave like microalgae. So we call them cyanohabs. But back in 2011, there were some massive blooms documented out in Lake Erie. In 2014, about 500,000 residents of Toledo went without water for several days because one of these massive blooms was blown over towards the Toledo shoreline, and they extract a lot of water in the Great Lakes for public drinking water and just general household use. And they found that it was contaminated with really high levels of microcystins, which is one of the worst cyanobacterial toxins. In the 2011 bloom, they detected anywhere from 0.1 to about 8 or 9, but some of their samples indicated that they had thousands of micrograms per liter of the cyanotoxin. So it was incredibly dangerous levels of this toxin. And these blooms persisted for many weeks as well. And this, you can see a boat. This is from a National Geographic depicting an aerial shot, again, cutting through the water there. And you would not want to come in contact with that water.
Or drink it either.
Or drink it, correct. So even in the marine system, although many of these harmful cyanobacterial types are freshwater phenomena, they can make their way through rivers into transport into the coastal ecosystem. And there was an event that occurred back in about 2008, I believe, where a bunch of sea otters were washing up dead in Monterey Bay. And when they actually did an autopsy on these animals, they found that their livers were severely damaged. And so that's one of the places where the cyanotoxins can produce a fatal effect, is by liver poisoning. And it looked like the cyanobacteria were coming down from these inland sources, being deposited on the shoreline and then incorporated into the filter feeding shellfish that the sea otters would then come along and feed on and would become toxified. So these were really extreme levels of cyanobacteria too. That's just a natural grab from this Pinto Lake area up here. Okay, so here's some of the nasty compounds. Microcystin is probably the most common one, and it impacts liver function. We've detected these in lakes here in Maine. Nodularin's another one, also impacting liver function. Anatoxin, it was initially detected back in the early 70s when an entire herd of cattle in Canada keeled over dead after drinking in a pond. And they initially didn't know what it was. They call it very fast death factor. And it was just this little tiny compound here that had contaminated the freshwater drinking supply and lost a whole herd of cattle. Cylindrospermopsins, liver function. Some of them will actually produce just a skin rash, or they'll give you food poisoning like effects. Saxitoxin is one of the most potent nerve toxins. Apparently, spies during World War II would carry little capsules of saxitoxin. If you were caught, you would pop one of these and it'd be all over. And this is one that produces the red tides that we have to deal with in the marine system here. So we're actually set up down at Bigelow to monitor for this and perform a FDA certified method for that. And then one of the newer ones on the block is BMAA. It's a non-essential amino acid, and it looks like it has effects in the nervous system. There's been some research tying it to higher incidences of ALS in some communities. The linkage, I don't want to say that it's a really tight linkage, but there's evidence suggesting it could be related. So needs a lot more work. But we've also detected this over in New Hampshire. So this is the main one that a lot of labs focus on. In fact, my lab at Bigelow is going to be set up in the next few weeks to start testing for this using an immunological test. It inhibits liver function. It interferes with these phosphatases, and ultimately it leads to phosphorylation of proteins in the liver, which causes cell death. If that wasn't bad enough, it's also a tumor promoter. So it's just a really nasty compound. And this is the LR microcystin is one of 140 types. It's the most common one, so it's what people typically try to measure. And they measure it by just detecting the immunological response to this functional group here. And I won't really get into it, but there are some problems with this immunology-based method. The reason that everybody uses it is that it's relatively cheap. It costs on the order of $200 or $20 a sample, whereas the more analytical, precise methods would cost upwards of $200. But it could determine whether this molecule was intact or if you were just detecting this free ADDA residue. Okay, so again, my work started in collaboration with Whitney and Denise in the Belgrade Lakes, and they have a great sampling scheme set up in Great Pond and Long Pond, and the target there was Gliotrichia. It's a really interesting cyanobacteria. It's filamentous, but all the filaments stick together and form these dense colonies. It actually looks a lot like a marine species that we used to encounter when I worked out in Bermuda. Other interesting thing about it, it overwinters in the sediments. It forms these aconites, which are like a resting stage. And so they'll spend the winter in the sediment. They can take up a lot of phosphate from the sediment. In the spring, they become buoyant. They rise up in the water column, and they start fixing nitrogen, and they've got basically an onboard supply of phosphate from the sediments. So again, they're sort of like their own little farms in terms of productivity. It's very conspicuous when it blooms, as indicated from that title slide, but you can't necessarily see it coming, is how I would characterize it. It can be very low abundance, and then all of a sudden, either by wind activity or wave activity, or a combination of the two, and then just the population exploding pretty quickly, you can see concentrations of it up against the shoreline. So our goal was to develop a genetic-based system to allow us to see it when it was at really low abundance. without necessarily having to do a lot of tedious microscopy. Little bit of history on Gliotrichia. It is a confirmed toxin producer. This is a report from back in 2017 by a group over at Dartmouth and UNH. And they basically picked out those colonies, picked out about 100 colonies or so, and measured essentially microcystin LR, that Maine microcystin type, in these 100 colonies each. The World Health Organization has set a limit for microcystin LR in drinking water of about 1 microgram per liter. Okay, that's free microcystin plus any that's tied up in cells itself. So it's a pretty low limit if you remember those values from Lake Erie that ranged generally from about 0.5 to about 9 or maybe up into the thousands. Gliotrichia is found all throughout New England. Many sites in Maine have it as well, but it can be in pretty low abundance in a lot of different places. Long Pond and Great Pond were areas where it did have a very high abundance. Notice Sabatis Pond down here. I have a slide on that in a minute. So the method that we developed, again with Whitney and Denise and some of their students, was based on extracting the DNA from these cells, so cracking all of these cells open, releasing all of that genomic material into a solution, and then developing specific DNA probes that could go in and find exact matches to that target species amongst thousands of other different types of DNA, and also all the other DNA that came out of the target cell. So we use these little short genetic probes that can go in and find binding sites and then essentially run off carbon copies of that little, oops, I just killed myself. Okay. Run off carbon copies of this little short region of DNA such that we can actually see those copies increase in abundance over a period of about an hour. So it's a rapid test to tell you, A, do I have it present, but also we can use this technology quantitatively to figure out how many gene copies there were. If we know something about how many copies one little cell in each of these strands contains, we can then estimate the total amount of Gliotrichia in a water sample without even looking through a microscope. So this is what a filtered sample looked like from Lake Winnipesaukee last summer. And typically that's what we're working with. And you just looking at that filter, you'd have no idea how many how many individual colonies, let alone individual cells there were. We did some initial DNA sequencing. This is going back to Great Pond and Long Pond. And then there's one sample from Lake Auburn here as well. And we confirmed that yes, these sequences that we were pulling out of our water samples did in fact match most closely with Gliotrichia from public DNA databases. And then we honed in even further. That first gene we looked at told us, yes, it's Gliotrichia, but it turned out there wasn't enough genetic variability in that one gene to discriminate Gliotrichia from other closely related species. So we had to move on to a different gene. The problem was there were no close relatives in the public databases for that other gene. So many of these cyanobacteria, although they've been around for about 4 billion years, we haven't characterized all of their genetics very, very well yet. This is the little short strand of DNA that we amplify, and then these are the little probes that we add into our sample to make these carbon copies of this short region. The short region is only, I think it's about 120 base pairs or so. And this is what it looks like on the computer screen. And so you start off with nothing there, and then all of a sudden the lights come on as the first sample starts to amplify. And as the cycling progresses, the DNA standards and the unknowns start to amplify throughout the cycle. So the standards that we run that have the highest DNA copy number start to come up first, whereas the lowest concentration standards come up last. and then we run unknowns, and then we relate these unknowns to our standards, and we can get an estimate of copy number. That's the basic principle. So you run a DNA standard of known concentration, you run your unknowns, figure out how many copies of that gene target there were in your sample. And when we did this for Great Pond and Long Pond, we got a bunch of graphs that look like this over a season. These are data from 2015, and you can see that early in the summer there's nothing happening, and then all of a sudden we get this big spike in the amount of gliotrichia. And the point is that you'd probably never even notice that gliotrichia were in your water in the early part of the summer. You definitely notice it up here when there's 50,000 cells per mil. And we did this for, again, 5 different sites in each of the lakes. We detected some very high copy number concentrations. Other times of the year, or the summer, it just wasn't there at all. This is same type of analysis for Long Pond. So again, it's a rapid technique. We ran all of these, once we got the method worked out, we ran all of these samples in one afternoon. So it's not like sitting there under the microscope and counting for days and eyesight going blurry. Another site I've worked at is Sabattis Pond, and this is one of these very green ponds. It's a very shallow pond, and many different types of cyanobacteria. In fact, the cyanobacteria were really the only thing I could see under the microscope. And this is just looking at the gene that we typically sequence. It's not particularly important, but it's just pointing out that we sequenced this one little short fragment of DNA within this gene. And we can do this. So this is a different technology that I'm talking about now. Now we're just taking all of that DNA from a water sample, grinding up the water sample, releasing all that DNA, and then just massively sequencing it. So for this one sample from Sabattis Pond, we generated 50,000 DNA sequences. And then we can compare all of those sequences to one another and find out, okay, how many different individual types did we find there? One of the major components was this group, Aphanizomenon. That's a toxin producer. So 45% of our DNA sequences were Aphanizomenon. We detected microcystis. That's where microcystin got its name. Many of these nostoc type of cyanobacteria will also produce toxins, as well as pseudoanabina. So Sabattis definitely is worthy of further investigation, and it's definitely one of the most diverse pools of cyanobacteria I've seen. Okay, so getting to the point now about what you can do and how you can contribute. I've started experimenting with some technology it's smartphone based, and it's driven by an app, and it's a tiny little machine that you can take out into the field. So this is last fall with a group of students from Colby College. We're out at Flagstaff Lake. So we hiked in 2 miles, collected a water sample right here. You can see we're actually standing in the water. We did a 5-minute DNA extraction at the end of the dock, and then we ran the genetic test with the little handheld device right at the end of the dock. Well, we went canoeing. We came back an hour later, and we had the results, and we detected gliotrichia at Flagstaff. So again, it's a rapid method, and it's relatively easy. If anybody wants to, they can talk to my son, who's a recent graduate of 7th grade, and I demonstrated this technology in his class a few weeks ago, and the kids seemed to understand it. They could relate to the app. It's a simple matter of programming the cycling, and then even the DNA extraction it's a 5-minute protocol that I could train all of you on in about 10 minutes.
So shouldn't you be standing in that if it's a toxin?
Well, it was very low abundance. I mean, we didn't see a bloom going on, but questionable. We don't know enough about it yet. And again, this is what our sample from Flagstaff looked like, and this big, strong exponential curve indicates a positive result for gliotrichia.
Did you expect that?
I didn't know. Had no idea. I talked to the folks at DEP, and they were not familiar with any reports of gliotrichia in Flagstaff. So it may have been a first sighting. This is what the technology looks like now. It used to be a slightly different form factor, but you can set it up and now run 9 samples. You program it again with your smartphone via Bluetooth, and then you close it up and let it run. This was a little test we did to determine, was it Atlantic cod or Pacific cod? But it's the only picture I had. So again, this is what I have set up is a little mobile DNA sampling unit. This is on the front porch of our board member's cottage over on Winnipesaukee. We filtered water samples down. We could do a quick DNA extraction. We have other technology at the lab and are hoping to invest in this kind of technology. This is the latest quantitative PCR-based technology that's, again, similar to the handheld, but this is a lab-based system, and found really good results between this newer technology and the technology we've been using. We've started doing some cyanotoxin measurements, and I'll just point out that these are natural abundance cyanobacterial samples from Sebatis. We detected 0.62 micrograms per liter of microcystin. Remember, the World Health Organization limit is 1. So we're getting close to the WHO limit at Sebatis. We also detected this BMAA compound in Sebatis. A net toe sample from Winnipesaukee, which is, again, a net toe is going to concentrate all the cells, so these numbers are going to be inflated over natural values. We detected a pretty high level of BMAA from the gliotrichia bloom. So we know, well, it suggests that glio might be producing BMAA. Hopefully we'll get some more confirmatory samples. I guess if there's any redeeming data here, it's that there was no free microcystin in the water from Winnipesaukee. It all seemed to be cell-bound. I mentioned that we do a bit of toxin testing at the lab, so I'm setting up for these quick, rapid, ELISA-based, immunology-based toxin tests, but we also have a lot of analytical capability, as does Whitney, I think, is developing some at Colby, and this gives us a higher degree of confidence that the toxins that we're measuring are what we think they are, and at concentrations that may or may not be of concern. So, in conclusion, Maine glio is highly similar to the glio in the database. We have a new method for detecting gliotrichia, and it appears to be accurate in terms of estimating cell numbers. This portable PCR is showing a lot of promise for citizen-based science, and that this newer technology, again, that we're hoping to invest in, agrees well with traditional quantitative PCR. The nice thing about this ddPCR for droplet digital PCR is you don't have to run the DNA standards that I was referencing. It uses statistics to calculate how many gene copies must have been in your original sample to get the level of amplification that you see. So what that does is it kind of frees up your analytical capacity. You can run more unknown samples and spend less time running your standards. And I'll just leave you with, this is a little GoPro video I shot. So this is why I was invited over to figure out what's that stuff growing underneath my dock. And as I snorkeled around here, it became clear that there was something else going on. So gliotrichia tends to concentrate right in the surface layer. So the top 10 or 20 centimeters or so is just thick with it. I shot this video, handed it off to our board member. He sent this to contacts in the state of New Hampshire. Within 48 hours, the state had issued a warning about coming in contact with the water here. It didn't make me feel good about snorkeling in it. So, if I hadn't shown up that day, they wouldn't have really known about this issue, and they wouldn't. Since then, they've actually issued bonds to investigate water quality in this part of the lake. They've set up a cyanobacterial task force, all because of a 2-minute video. And so there's a lot that we can do as citizens and scientists to help with detecting these rare events. A week or two later, this bloom was gone, and you'd never even know that it was there. But it probably had left some residual toxins in the water. There's unknown ecosystem effects. I guess I'll just leave it at that. Yep.
So with the high concentration of the gliotrichia toxin, is it mainly going to be affecting the liver in organisms, fish, humans, whatever, or what else could be affected?
I mean, right now we know what it does to vertebrates, I guess. We don't necessarily know how else it might be affecting the food web. We don't really know if it's showing up in the fish. Presumably, if they're feeding on a lake food web, that toxin will get passed up through the food chain. But nobody's making these measurements yet.
Did you test this stuff with your little handy dandy thing?
We did, yep. I mean, the nice thing about gliotrichia is that when it looks like this,
Pretty obvious.
it's pretty obvious. You can identify it as gliotrichia, but just to make sure the assay that we developed with Maine gliotrichia was going to work, because you don't always know that. That's a good question. I mean, you develop a new assay for one part of the world, and then you transfer it to somewhere else, and it may or may not be genetically close enough to react. It did react.
And so what was all the sort of stuff on the bottom?
I don't know. I got distracted by the cyanobacteria.
Metaphyton.
Yeah, but it wasn't green.
No, it's that cloudy
Metaphyte.
flocculent material.
That's what metaphyton does when
It
it
usually
goes
looks green, not look white.
over.
How affordable is the little portable?
The portable device, I believe they're selling it, the new version of it, which runs 9 samples for $6,000 is the base price. It can, without getting too technical, it can detect up to 3 different colors simultaneously, and if each level of detection is another $1,000. But it's, for under $8,000, you can do genetic testing at your dog. So it seems like it's the kind of technology that could be affordable to, a lake association.
So you mentioned that you don't always see these blooms coming.
That's right.
But if you're tracking this data with, say, your system and have this idea of what's going on, could you then correlate, say, the appearance of a sudden bloom with a 2-week-long temperature spike?
Yeah, I mean, that's what we would love to do. We'd love to start, using this technology in coordination with existing sampling efforts and even the drone type of work. It's really cool. I've talked to a group over at Dartmouth that's doing some hyperspectral lake mapping, and they got really excited about trying to do some on-the-ground sampling while the drone was flying over. So,
I'd like to thank Dr. Conway
Yeah.
for his presentation.